Okay, so good morning to all of you. Today we are taking topic bacterial ultrastructure. Okay, we have already discussed about cell wall of a bacteria. Now I'm taking the ultrastructure of a bacteria. What are the different organelles present in bacteria? You can categorize or you can classify bacterial ultrastructure in many ways. One is on the basis of their growth requirement or on the basis of survival, essential organelles and non-essential organelles. Likewise, we can categorize the bacterial ultrastructure, the organelles present in bacteria on the basis of its location or position. Okay, so this picture you have already seen. This is the shape of a bacteria where you can see different organelles present in the bacteria. So there are some organelles which are present outside the cell. These are external to the cell, like for example capsule, right? Like flagella, or this you can see this structure, pilus, this structure fimbriae. So fimbriae, pilus, flagella, capsules, these are present outside the cell wall. These are external structures. So we'll discuss all the organelles in brief one by one. So again, for information, prokaryotic cell, it is divided into external, then cell envelope, then internal structure. So external structure includes appendages, for example, flagella, pili, fimbriae. Likewise, one more layer present outside the cell wall, which is glycocalyx, we will discuss today. So, at a glance, these are the different organelles present in the bacteria, and these are given their functions. For example, plasma membrane. You will learn more about plasma membrane in cell biology class, which is a selective permeable barrier, right? And which allows the transfer of nutrients and waste, and involves in the detection of environmental cues for chemotaxis. Likewise, gas vacuoles are present. These gas vacuoles helps in uh, floating of bacteria. It provides buoyancy in aquatic bacteria. Those bacteria which lives in aquatic system, it provides buoyancy. Likewise, ribosomes, which are present as 70S in prokaryote, subdivided into 50S and 30S, it is a site of protein synthesis. Likewise, there are so many inclusion bodies or reserve deposits present in bacteria, and which helps the bacteria in unfavorable conditions by providing different nutrients required for the bacteria for survival and growth. So these are known as inclusion bodies. Likewise, nucleoid, the genetic material, where the genetic material is present, nucleoid. Then periplasmic space, we have already discussed, it contains so many enzymes. Cell wall, you know about this thing. Capsule and slime layers. Today we'll discuss about capsules and slime layers. Then fimbriae and pili, which helps in adherence. Pili helps in conjugation. Flagella, which helps in movement, and endospores, which help in survival under harsh conditions. So these are the organelles present in bacteria at a glance, and these are the very brief function of these organelles. So today we are talking about external structures, which includes flagella, pili and fimbriae, capsule and slime layer. So we'll take one by one. Okay. So again, I am requesting you to confirm whether you can see this slide. Glycocalyx, EPS,
Yes,
ECP. Okay. Can you see this?
Yes, sir.
yes, sir.
Yes, sir.
Okay. Am I audible?
Yes, sir.
Fine. Absolutely good. Okay. So in bacteria, outside the cell, there is one more layer present, which is known as glycocalyx or EPS. EPS stands for extracellular polymeric substances or extracellular polymers. So extra polymeric substances means these are the polymers, right? These are the polymers, and polymers of what we'll discuss. These are the extra polymeric substances which are present outside the cell wall. Cell wall के बाहर पाया जाने वाला एक ऐसा लेयर जो कि polymers हैं, उनको हम in general we are calling it as a glycocalyx or EPS or ECP. So capsule is basically made up of either polysaccharide. In some bacteria it is present as polysaccharide, or in some it is polypeptide. So polysaccharides means it is made up of sugars, repeating units of sugars. Now these repeating sugars may be same sugars known as homopolysaccharides, or maybe of different sugars, then it is known as heteropolysaccharides. So capsules which are present outside the bacterial cell wall, it may be made up of polysaccharides, which can be homopolysaccharides or heteropolysaccharides. It may be made up of proteins known as polypeptides, right? So capsules can be divided on the basis of composition as polysaccharides or polypeptides. Likewise, capsules on the basis of dimension, if it is more than 0.2 micrometer, it is known as macrocapsules, otherwise microcapsules. So it may be macro capsule or it may be microcapsules. Now actually this capsule, which is a part of glycocalyx, can be present in two forms. You can see this picture. Here in this picture you can see the outer layer present outside the cell wall, which is well organized and not easily washed off. यानी कि एक ऐसा लेयर जो कि थिक है, स्ट्रक्चर्ड लेयर है. You can see the structured layer which is not easily washed off, then it is known as capsules. But if it is loose gel-like structure, you can see this, which is not well organized and which can be easily washed off, then it is known as slime layer. So the extracellular polymeric substances or glycocalyx can be in two forms. One is capsule, this is capsule, or this is capsule, or slime layer, this is slime layer. So capsule which is thick, structured, not easily washed off. Yeah, I have taken the reference from Prescott and Talro. Yeah. In case of slime layer, it is not well organized, it is loose, it is gel-like structure, and it can be easily washed off. You can see this picture, which is a staining micrograph of capsule of Klebsiella pneumoniae. Yeah. So this is a ultrastructure of Klebsiella pneumoniae. You can see this structure, right? But in case of archaea and some of the bacteria, there is present in a form of glycoproteins, which is known as S layer or RS layer, and it appears as a flue tiles-like pattern. So in archaea and in some bacteria, it is known as S layer. So the glycocalyx or extracellular polymeric substances or extracellular polymers, it can exist as capsules, it can exist as slime layer, or it can exist as S layer or RS layer. The function of this capsule is basically binding or adherence to the surface or the substrate. It also gives pathogenicity, means disease causing ability to the bacteria. It has a medical significance. It also provides anti-phagocytic property. What does it mean? Anti-phagocytic property means the bacteria which is capsulated is not engulfed by macrophages. Macrophages, which are the part of our immune response, it cannot phagocytize the bacteria containing capsules. So it has an anti-phagocytic property. It also helps in desiccation, prevent desiccation, and provide nutrients to the bacteria. Coming to next slide. The next is the appendages which is present outside the bacterial cell wall. The very common is flagella, right? So flagella is the organ of locomotion in motile bacteria. So in motile bacteria, the organ which helps in locomotion is flagella, right? Flagella, which is a web-like structure and which is 15 micrometer long and 20 to 30 nanometer in diameter. So the length of the flagella as well as the diameter of the flagella varies in bacteria, right? But it is generally present in all motile bacteria. Note point is due to the surface area or Reynolds theory, flagella is common or generally present in bacilli or bacillus or rod-shaped like bacteria. In rod-shaped like bacteria, flagella is present while it is generally absent in tocci. So the number of flagella and the position of a flagella varies in bacteria. यानी कि फ्लैजिला की नंबर और उसकी पोजीशन वेरी करती है एंड यू बी कॉल्ड इज अ फ्लैजिलेशन, द डिस्ट्रीब्यूशन ऑफ फ्लैजिला इन बैक्टीरिया. So if flagella is present in a bacteria, we call it as a trichus, and if it is absent, it is called as atrichus. So trichus means the presence of flagella. Now the flagella may be present at the poles, and if it is present at poles, then it is known as polar flagellation. It is called as polar flagellation. If it is distributed all over the surface of the bacterial cell wall, then it is known as non-polar flagellation. मैं क्या बोल रहा हूँ? मैं बोल रहा हूँ कि अगर फ्लैजिला प्रेजेंट है बैक्टीरिया में तो हम कहेंगे ट्राइकस, नहीं है तो कहेंगे एट्राइकस. फ्लैजिला की पोजीशन या लोकेशन कहाँ पर है? फॉर एग्जाम्पल ये बेसिलाई है, ये दिस इज, यू कैन सी दिस डायग्राम, right? This is a bacilli and you can see the flagella which is present at one pole, more than one flagella at one pole. So if it is present at poles, then it is known as polar, but if it is present all over the surface, then it is non-polar. Now depending upon the position of flagella and number of flagella, what I am saying, on the basis of position of flagella and number of flagella, it can be further categorized as monotrichous. Monotrichous means, mono means one, trichous means flagella. When one flagella is present at one pole of a bacteria, then it is known as monotrichous. You can see this picture here. This one flagella is present at one pole. This is known as monotrichous. Examples are Vibrio cholerae, right? And some species of Pseudomonas. So monotrichous, one flagella at one pole. Amphitrichous, flagella which is present on both poles, like Alcaligens facialis, amphi on both poles. So flagella which is when present at both poles of a bacteria, then it is known as amphitrichous. Then cephalotrichus. Cephalotrichus. When more than one flagella present at both poles, like Pseudomonas fluorescens. Lophotrichus. Here you can see this Lophotrichus. When more than one flagella are present and at one pole, Lophotrichus. Example is Parallum volutens. Right? Peritrichus. Peri means all over the surface. When flagella is present all over the surface of bacterial surface, then it is known as peritrichus. Example is Proteus vulgaris. So, polar flagellation can be monotrichus, amphitrichus, cephalotrichus, lophotrichus, peritrichus. Coming to the structure or components of a flagella. So, flagella is generally made up of three main components. One known as basal body, this is basal body, hook, and this filament. So flagella is made up of three main components: basal body, hook, and filament, right? But the structural organization of flagella varies in gram negative, this is gram negative, and gram positive. Here you can see this is plasma membrane and this is peptidoglycan layer. Okay, while in this case this is plasma membrane, this is peptidoglycan layer, and this is outer membrane. So in case of gram negative bacteria, there are four rings. You can see this ring like structure in basal body. These rings are called as L ring, P ring, S ring, and M ring. L, P, S, M. There are four rings present in gram negative bacteria. And you can see this diameter is 20 nanometer, 22 nanometer. So in case of gram negative bacteria, four rings are present, L, P, S, M, followed by hook, followed by filament, while only two rings are present in gram positive bacteria. So there is a difference in structural organization. Okay, so about the motility or movement, how a bacteria moves with the help of flagella. So in general, we can say it is like a turbine motion or movement, right? This is a flagella. You can see this is a flagella. This is a bacteria. Sorry, this is a bacteria, right? And with a flagella. So when this flagella moves anti-clockwise, when this flagella moves anti-clockwise like this way, then the bacteria moves forward. What I am saying, if the flagella moves anti-clockwise, then bacteria moves forward. But if the movement of flagella get reversed, means if it moves clockwise, then it stops or tumbles. So either it can move forward, bacteria can move forward, or it can stop, right? It will not move backward. Either it will move forward or tumble. So it depends upon the movement of flagella, whether it is anti-clockwise or clockwise. Here you can see this is a single flagella. But in case of peritrichous bacteria, means if the flagella is present all over the surface of the body, now you can see all flagella, means even each flagella moves anti-clockwise, and it helps the bacteria to move forward. Likewise, if each flagella moves clockwise, then it tumbles or stops. So this is how bacteria moves, and the energy which is required or involved in the movement is proton motive force, PMF, which is H plus ion movement. So proton motive force is involved in the movement of the flagella, means it gives energy. So here this is showing a picture where you can see the flagella, all the flagella which is moving anti-clockwise, then it is run. But if each flagella moves clockwise, then it tumbles. One more important property in bacteria is seen that the bacteria moves not randomly. Okay, it moves towards the attractant, and here the attractant is obviously some kind of energy source or nutrients. So if the energy source or the nutrients is chemical in nature, then the movement of bacteria is called as chemotaxis, means movement towards chemical substances. If the movement of bacteria is towards light, then it is known as phototaxis. So the movement of bacteria with the help of flagella is not a random one. It is well organized and it moves towards a particular direction, towards either a chemical substances or towards light. So it is called as chemotaxis or phototaxis. Now this picture is showing or depicting the bacterial movement towards chemical. Now you can see here, this picture is showing the bacterial movement and tumble is very short, means run length is very short. Here this is short runs and it is random followed by a tumble. So the red is tumble and green is run. So green is run, then tumble, then run, then tumble. So you can see that there is a short run. But here, if you compare this picture, you can see that there is a long run. The bacteria moves from this position to this position, then tumble, then moves from this position to this position, then tumble, then movement. So you can see this long run, more time on run. This happens when the bacteria is moving towards the attractant. Attractant, attractant means the chemical substance which is guiding the bacteria to move towards the chemical substances because it will be a source of energy. For example, if glucose is present in a medium, the bacteria move towards glucose. So here glucose is attractant. In other case, if it moves away from the chemical substances, then that chemical substance is said to be repellent. Right? So means the bacterial movement towards chemical substances. This is the pattern how the bacteria moves with the help of flagella if the chemical substance is attractant. Coming to next slide. Lab demonstration. How you can demonstrate the motility of a bacteria in lab. Okay. First I would like to show a video, and please confirm me, CR can confirm me, can you see this video or not?
Yes, sir. It's loaded.
Can you see this video? क्या वीडियो देख पाए?
Yes, sir.
This video is showing the bacterial mortality. Fine. So we can demonstrate the mortality of a bacteria in laboratory by three methods. One is known as wet slide method. Wet slide method where there is no fixation of bacteria onto the slide, right? And we observe the bacteria directly under the microscope, which is called as wet slide method. Then another method is hanging drop method. I will try to show you a video of this method, a hanging drop method, where bacterial culture or the suspension uh, drop hangs onto the slide. There is a special slide known as cavity slide, and we use uh, this hanging drop method to demonstrate the mortality. And another method is a method where we use semi-solid agar medium. Semi-solid means where the concentration of agar is basically a solidifying agent, right, which is added to the medium. Here the medium is basically the nutrients which is required for the growth of bacteria. So we add agar, the concentration of agar is less and the medium becomes semi-solid, solid, partially liquid. And in this case, you can see the mortality of a bacteria. Okay, can you please again confirm me, are you able to show this slide where it is written mortality test, media results?
Yes, sir.
Right.
Yes,
Okay.
sir.
So you can see this picture. ये देख रहे होंगे इस पिक्चर में. This is non-motile. ये यहाँ पर आप देख रहे हैं कि ये ट्यूब्स हैं, right? आप सिर्फ assume कर लीजिए this tube is containing some media, right? And the bacteria is inoculated or transferred into this media. Bacterial culture is transferred into this media. Now, if bacteria is non-motile, then what will happen? If bacteria is non-motile, then the bacterial growth will occur on the line of inoculation. ये जो आप लाइन देख रहे हैं, ये लाइन बेसिकली लाइन ऑफ इनोकुलेशन है. लाइन ऑफ इनोकुलेशन समझते हैं क्या है? ये टेस्ट ट्यूब है, इसमें मीडियम है. नाउ विद द हेल्प ऑफ inoculation loop or needle, basically, we transfer the bacterial culture into the medium, right? At the center of the medium, we call it as a stabbing. This is called as stabbing. The English word stab. So what happens after incubation? Incubation is bacterial. This bacteria is allowed to grow at 37 degrees centigrade for a required time, which is generally 12 to 24 hours. You can see the growth of the bacteria as a thread-like structure. But if the growth of a bacteria is seen like this way, means on the line of at the line of inoculation as well as on both sides of line of inoculation, like this way, then it is said to be a motile bacteria, because a medium is semi-solid and bacteria, because of semi-solid medium, bacteria moves on both sides of the line of inoculation, and this is how the media tube appears. Right here you can see exact single line of inoculation, the growth of a bacteria on this line only, but here it is somewhat distributed. So this is motile bacteria. So this is known as motility test using media. One of the very common media is sim agar medium, which is used for demonstrating motility of a bacteria. Right? So if it is asked, can you demonstrate the motility of a bacteria in laboratory? Or if it is asked, if a bacterial culture is given to you, can you check the presence of flagella in bacteria? You cannot see the flagella in simple microscope or this oil immersion microscope. You can see the flagella under only electron microscope, but you can demonstrate the presence of flagella by different slide test and culture test. Again, I am showing motility of a bacteria. Please confirm, are you able to see or not? This is connecting to a YouTube link where there is an original picture of bacterial mortality. Please confirm when it gets started. Are you able to see this video?
Yes, sir.
Okay.
Yes,
So
sir.
if you can see uh, you can see some transparent cells like structure and they are moving. I will show you. Yeah, this is basically a video taken uh, through objective lens under this oil immersion microscope. You can see this motility. कुछ छोटे छोटे ऐसा लग रहा है जैसे कीड़े हैं and they are moving, right? Can you see this transparent cells in your mobile
Yes,
or
sir.
in your system? Yeah, so
Yes, sir.
these are basically the bacteria which are moving. Basically, this is a slide, right? This is a slide, and the slide is showing the bacterial movement, and you can see this movement. This is very, very fast, and the bacteria are basically in the log phase of their growth. Means they are young. You can say, वो बिल्कुल young है bacteria, and the nutrients are there and they are moving fast. So you can observe this kind of movement under the microscope. Imagine it as a objective lens of our oil immersion microscope, and you are observing the cells. If you can apply stain to the cells, and you can see the cells which is observing as a transparent, you can see the cells colored as well. आपको colored cells दिखाई देंगे when we come to the laboratory after this lockdown, hoping that I can show you so many things related to this mortality, very interesting pictures of this movement. So these are all bacilli which are moving with the help of flagella, right? And you cannot say it's a monotrichous and bitrichous because you cannot see the flagella, but you can see only the movement of a bacteria. Yeah, so this is how we are showing this picture. So this was about the demonstration of flagella in laboratory. How you can demonstrate flagella? You can observe flagella under the microscope, but you can demonstrate the mortality of a bacteria by wet slide method, hanging drop method, and a semi-solid agar medium test. So this is all about the today's lecture. So if you have any query, you can ask me. So this is query session. You can ask me if you have any query. So today we have discussed about the capsules and appendages, which is flagella. In the next lecture, we'll discuss fimbriae and pili, another appendage which is present in bacteria. So if you have any query, you can ask me. You can raise your hand and you can ask if you have any query. I will share this PPT once it is completed. So this is from Unit 2 again, and we are discussing about ultrastructure of a bacteria at a glance, a brief about different organelles present in bacteria, structure, composition, and function. Yeah, yeah. One student is asking question. You can ask me.
What was the that helps the bacteria
What
to
was
move towards the
what?
attractive medium? What was the
Uh,
force that helps the bacteria to move towards the medium? So
Basically,
force.
yeah. Basically, it is actually proton motive force, right? There is a movement of hydrogen ions, which is produced inside the cell. There is a biochemistry. PMF force is there, and PMF force propels the flagella, right? and if it moves anti-clockwise, then it pushes the bacteria in a forward direction, and it moves the bacteria. So this is how bacteria moves. Now this motility uh, is of different purpose. For example, a bacteria which is living in aquatic system, it comes towards the light, right? With the help of its flagella, or it moves towards the substrate with the help of flagella. It may be a chemical substance. So the force behind this movement is proton motive force, PMFs.
Okay,
Yeah.
sir. Thank you.
Any other query? No query? Okay, fine. So I am stopping this lecture. If you have still any query, you can ask me in next lecture. Before starting the lecture, I will take queries first. You can note the queries or questions on the notebook, and you can ask me before starting the lecture. Okay?
Okay, sir.
Okay, fine. Thank you.
Thank you, sir.
Thank you, sir.
Thank you, sir.